हॅलो, आवाज येतोय माझा?
Yes, sir.
So if you see the difference between the dicot and monocot, epidermal cells are present in dicot, epidermal cells are absent in monocot. That is called as we call as a trichomes here. This hypodermis is made up of a cholenchymatic tissue in dicot. Hypodermis is made up of sclerenchymatous in monocot. Here endodermis is absent. In endodermis, endodermis is present in this dicot. Endodermis absent in a monocot. If you see ground tissue differentiated in the stellar and extrastellar zone, where ground tissue is not differentiated. Here, that entire sections we can find out the ground tissue up to this hypodermis to the pith regions here. Here in the dicot here, vascular bundles are limited in number. We have few numbers we can find out, but in the monocot, vascular bundles are numerous. Numerous. Another difference in vascular bundle, they have arranged in a ring, while in monocot, vascular bundles are scattered. If you see vascular bundle in dicot, these are the co-joint, co-lateral, and open. But in the monocot, these vascular bundles are by that is a co-joint, collateral, and closed type of a vascular bundle. Next, if you see, due to the why we call as open, because of a cambium are present in dicot region. In monocot, cambium are absent, so vascular bundles are the closed. Next, if you see here, in dicot, protoxylem is absent, while lacuna are absent, while in the monocot, this protoxylem lacuna are present. In the dicot, pericycle present. In this monocot, pericycle absent. Bundle cap present. Bundle cap absent. So bundle seat present. Yeah, is in the dicot. Bundle is absent. While this in monocot, bundle seats are present. Here in the phloem tissues here, phloem parenchyma and phloem fibers are present. While in the monocot, phloem parenchyma and phloem fibers are absent here, because if you see this is the not tall except the bamboo. In monocot there is nothing the taller plant. Okay, very shrubby habitats we can find out. So this is the reason here. In this dicot here, pit region present. In monocot, pit region is absent here. Again in the pit regions we can find out the medullary rays, while in the monocot medullary rays absent in the monocot. And here due to the presence of a cambium here, this having the secondary growth, while due to the absence of a cambium ring in the monocot, there is a lacking of a secondary growth here. So this is the main difference in between the monocot and dicot. तर तुम्हाला अजून जर याच्यामध्ये माहिती लागत असेल तर तुम्ही इझी बायोलॉजी क्लास ऑनलाईन वरती गेलात तर तुम्ही इथं तुम्हाला बरेचसे अजून पीपीटी तुम्हाला याच्या रिलेटेड बघू शकता. ओके, नेक्स्ट आपण याच्यामध्ये काय प्रॉब्लेम, अनाटॉमी ऑफ मोनोकॉट काय रिपीट करायचं एखादा पॉइंट. डर्मल टिश्यू वरती, हायपर टिश्यू मध्ये, ग्राउंड टिश्यू मध्ये, व्हॅस्क्युलर टिश्यू मध्ये काय प्रॉब्लेम असेल सांगा, परत एक्सप्लेन करतो. कारण आपल्याला अजून सेकंड सेमचे प्रॅक्टिकल स्टार्ट करायचे, थेरी स्टार्ट करायची. काय प्रॉब्लेम आहे का? येस किंवा नो? Okay, please see the second portion here, that is a cortex region. Firstly we see outer cortex. So if you below this epidermis, so we can find out here some kinds of a greenish tissue. इथे दिसतंय ना, greenish tissue दिसतंय. Okay, green color. That means these are some kinds of a chlorenchymatous. Chlorenchymatous means they have the presence of a chlorophyll pigment, and these are used for the photosynthesis. So they are here. Also, we can find out the loosely packed cells with having the interstitial spaces. In the xerophytes here, this cortical cells forms the palisade-like tissue. जसं आपल्याला असं बॅरल शेप लॉंग पॅरल सेल अरेंज असतात त्याला आपण पॅलिसाइड टिश्यू म्हणतो. या झिरोफायटिक प्लांट्स मध्ये आपल्याला सापडतात. So what is the function of this cortex region? Is for the photosynthesis here. So we can find out here this kinds of a tissues here. So this is called as the outer cortex or cortex region. Next, if we see inner cortex. So below this outer, this here large cells we can find out. This large cells nothing but the we call as the inner cortex. So tissue inner to the outer cortex here. Next, these cells are made up of a parenchymatous, loosely packed cells. इथं loosely तुम्हाला दिसत आहे. Irregular असल्यामुळे काय होतं याच्यामध्ये space intercellular space create होतं. आणि what is the function? Is the function for the storage of food material that is called as a carbohydrate. So this inner cortex they have we can find out here. So next here, in some hydrophytes there is some kinds of adaptations we can find out. So in the hydrophytes, in the cortex regions, we can find out this some kinds of parenchyma cells here. So here in hydrilla, in lymphia, we can find out these kinds of adaptations here. And we can here large empty vacuoles we can find out is nothing but the cortex regions, and here's the storage of gas here. So there is a buoyancy are created. That is a buoyancy. That means the plant tarafta. So due to these adaptations. So this parenchyma helps the exchange of gases, as also it's provide the buoyancy to the plant. Next, if you see in the secondary, some kinds of a dicot like the eucalyptus, eugenia, ficus, the parenchymatous patches we can also observe in the cortex here. So here, yeah, as like this here, and reason canals occur in the cortex of anacardium. Yeah, एवढं डिटेल्स नाही, फक्त तुम्हाला स्ट्रक्चर बघून घ्या फक्त. ओके, समटाइम्स लॅसिटिफायर सेल्स ऑकर इन द कॉर्टेक्स, दिस प्रोड्यूस द लॅटेक्स. जर तुम्ही E4B फॅमिली बघितलं तर ह्या कॉर्टेक्स मध्ये ह्या टिश्यू असतात, लॅसिटिफायर्स टिश्यू, आणि त्या काय ज्यावेळेस इंजुरी होते तर तिथून काय होतं, लॅटेक्स काय करतो, बाहेर पडतो. ओके, नेक्स्ट द फंक्शन्स ऑफ द कॉर्टेक्स, इट्स प्रोव्हाइड द मेकॅनिकल सपोर्ट. During the secondary growth, hypodermis give rise to cork cambium, and there is a formation of bark. This cortex, especially, is made up of a collenchyma cells in the outer cortex can do the photosynthesis, while the inner cortex it is made up of a parenchymatous cells. It is used for the storage of food material, that is called as the store the carbohydrate. Next, cortical cells also store the egastric substance like some kinds of egastric means some kinds of chemicals that plants have, like the resin canals, latex canals, latex, resin, as like this. Okay, and also some plants we can find out the resin canals and latex canals here. So next to the if we see here, second is the cortex, and next is the endodermis here. So if you see here, this one, इथे अशी layer दिसते बघा. So हे आता इथं upper outer, inner, आणि इथे endodermis. So innermost layer of cortex इथे आहे innermost last layer, म्हणजे epidermis खालून ते अप टू द एंडोडर्मिस आवडा रिजन्स कोणता आहे, कॉर्टेक्सचा आहे. ओके, म्हणजे आउटर आहे, इनर आहे, आणि त्याच्यानंतर हा इथंच बघा तुम्हाला डायरेक्टली डिफरन्शिएटेड होत आहे. अशा या स्मॉल सेल्स तयार झालेल्या आहे, आणि त्याचा कलर कसा आहे, जे स्टेनिंग केल्यानंतर तो रेडिश सेल होतो, आणि कॉर्टेक्सचा तुम्हाला व्हाईटच दिसतो. So that is a distinguish. You can find out again this part that is called as the endodermis, innermost layer of a cortex. Very distinguished in a pteridophyte. Not distinguished in gymnosperm, angiosperm मध्ये आपल्याला याच्यामध्ये distinguish सापडत नाही, कारण this is the starting. pteridophyte मध्ये endosperm endodermis तयार झालेले आहे. Okay, so if you see, endodermis cells accumulated the starch grain. Okay, starch grain, hence is called as the starch silt. म्हणून त्याला काय म्हटलेले आपण, जो काय व्हॉट एव्हर द स्टोर्ड फूड मटेरियल आहे इथे जास्त स्टोर होतं, म्हणून त्याला काय म्हणलं, starch silt. If distinct endodermis is uniserate with barrel septa, असे एकच लाईन असते, सिंगल लेअर, आणि जसं एंडोडर्मिस दिसतं तसंच हे काय तुमच्या इपिडर्मिस जे जसं सेल्स अरेंजमेंट आहे तसंच तुम्हाला इथे पण दिसतं, बॅरल सेप्टा. सो असं या टाइप मध्ये स्टार लाईक त्याची ग्रोथ असते. So that's we call as some kinds of endodermis region. These cells are especially made up of parenchymal tissue and it is a closely packed. So here we have detailedly seen here these kinds of cell, and again here there is some kinds of again different kinds of casparian bands we can find out. जे हायर सेकंडरी ग्रोथ होते जास्त वाढ होते तिथे तुम्हाला ह्या टाईपचा कॅस्पिरियन स्ट्रिप सापडतात थिकनिंग साठी. ओके, इफ यू सी they have the characteristic thickness radial and intertaginal wall. They have called the casparian thickening. It is also called as the casparian bud. हे तुम्हाला काही गरज नाही, पण तुम्ही जर बघितलं असेल आपण सागाचं लाकूड, साग, टिक्टोना ग्रँडिस, अॅकेसिया निलोटिका, हे लाकूड जास्त मजबूत मजबूत असतं. का असतं? ड्यू टू द प्रेझन्स ऑफ दिस काइंड्स ऑफ casparian band, and they have secretes a particular kinds of a secondary metabolite that is called as the suberlin and lignin, and they have deposited inside the cells. त्याच्यामुळे त्याची काय होते लाईफ ड्युरेबिलिटी वाढते ड्यू टू दी दिस लिग्निन अँड सुबरलिन. ओके, बोथ आर दी इम्प्रेसिव्ह टू वॉटर. हे वॉटर इम्पर्क्युलेट करत नाही म्हणून ते लाकूड जास्त. ओके, so back passes of water and solid through the protoplast and endodermis. So this is the function of endodermis. Exact function of endodermis not known. पण आपण असं म्हणू शकतो ते पिथ रिजन आणि हायपोडर्मिस तो सेपरेशन करतं ते. It's not allowed the passage of water and not can store the food material as the charge grade. So next we want to see the still region. So if you see this epidermis, then we see this cortex. We also see this here endodermis, and this region below this endodermis up to the pith region, that we call as a still region. याला काय म्हणतो आपण still region. ओके, कुठून पासून? एंडोडर्मिस पासून ते पिथ पर्यंत. सो दिस इज द सेंट्रल व्हॅस्क्युलर सिलेंडर. सो दॅट इज द सेंट्रल पार्ट ऑफ द वूड. कुठे आहे? सेंट्रल आहे. So it's composed of again the four parts: pericycle, vascular bundle, medullary rays, and pith. So after below this endodermis, the pericycles are present. These are the vascular bundles, xylem and phloem. So this one, yeah, we can clearly find out here. Some kinds of larger cells, as the protoxylem, metaxylem, and as well as we can find out the dark stain cells here. This is nothing but we call as the phloem tissues here. Yeah. So this is the one vascular, second, third, fourth, fifth, sixth, seventh, as like this here. And if you observe here, these are arranged in a circle, in a ring. Okay. So this is we call as some kinds of a pith or still region here. Okay. So we see one by one here. So this here. So this started from below the endodermis, outer layer of still here, that is called as a pericycle, located next to the endodermis, show wide variation. Plant to plant, it's having the variation. In some plants, this pericycle is totally absent. Usually, it's having the multi-layer up to the 3 to more. If you see here, we can find out here 1, 2, 3. Okay, so this one. So if you see, this pericycle is made up of completely parenchymatous, or some kinds is also having some kinds of sclerenchymatous or alternating band. काही मध्ये तुम्हाला parenchyma आणि sclerenchyma दोन्ही सापडतात. So this is the still region. So here next we see vascular bundles. So this is called as the fascicles. These are vascular bundles. So this is the diagrammatic view. This is the slide view. So diagrammatic view, there is a sketch we have created, this one. So these all are the vascular bundles, and these are the in the slide here. So located inner to the pericycle, developed from the procambium. Number of vascular bundles 6 to 8. 1, 2, 3, 4, 5, 6. So as like this here, 6 to 8 vascular bundles find out in the dicot here, arranged as a broken ring. असं रिंग आहे पण ती कशी दिसलेली आहे? ब्रोकन झालेली, but it can hold by the cambium here. So vascular bundles composed of a xylem and phloem. Xylem is placed inner to the cambium, while phloem placed outer to the cambium. If you see here, Hello. If you see here, so here is the some kinds of a cambium here. This large are the xylem, and these are the this outer side there is having the phloem region. So in between the xylem and phloem, there is the presence of a cambium meristematic cells, and that are responsible for the secondary growth here. So detailed structure we can observe here. So here is the larger same here metaxylem, protoxylem. These are the phloem tissue here. So these are the vascular bundles we see one by one. So this vascular bundle are co-joint. That means xylem and phloems are together here, open because of there is a presence of a cambium here. Enlarge means protoxylems are inner side, so that's called as enlarge. Collateral and or bicollateral. Okay, collateral and bicollateral. These are the different things here. ज्याच्यामध्ये जास्त सेकंडरी ग्रो होते, सेकंडरी ग्रोथ होते, त्याला आपण त्याच्यामध्ये बायकोलॅटरल मध्ये सापडतात. You see here, collateral usually the types one patch of xylem and one patch of phloem. The bicollateral मध्ये a medium patch of xylem lying with the two phloem patch, especially in the cucurbitaceae. We can find out this kinds of a structure. तुम्हाला एवढं डिटेल नाही, फक्त झायलेम सेल्स कशा प्रोटो फ्लोएम सेल्स कशा ते बघू आपण. This is vascular bundles, and we can find out this xylem here. The larger one we call as a metaxylem, smaller one is called as a protoxylem. Protoxylem inner to the pith region, where the metaxylem towards the phloem regions. Okay, so what is the main function of xylem? It is used for the water and mineral conduction. Okay, especially these xylems are made up of tracheids, vessels, fibers, and xylem parenchyma. So these are the two types. That's the proto and meta. So if we see the larger one, these are the metaxylem, while these are the smaller one, this we call as a protoxylem. So why we are differentiated one? So this is the originated firstly, so it get matured, and this is the young one. So this is a smaller one. So this is mature one is called as a meta, while the earlier one that is a young one we call as a proto. So there is also its form a part of xylem arranged toward the center, very less amount in the tertiary elements, large amount of parenchyma. So we see here, sorry. So here, next, if you see this form the protoxylem, more opposite to here, more trachary elements and protoxylem. Cells are large lumen. इथे तुम्हाला दिसतंय large as compared to here. So what is the function here? Conduction of water, conduction of mineral, providing mechanism support. So here, so phloem tissue. So it's having the it's composed of sieve tubes, Campion cell, parenchymatous cells, and these are the fiber here. It's also having the two types of phloem here, protophlloem and metaphlloem. Same here. That is maturity and before the maturity, the smallest cells is called as a protoxylem. Protophlloem here. So you can find out here. Yeah, this is the xylem, and this is the smallest, some kinds of a stem cell. These are the phloem region here, and the smallest we call as a proto, while largest one we call as a meta. Next here, so here, there's the cambium. This is the differentiated point of monocot and dicot here. So this cambium here. So we can observe here, this here. So here tissue आहे, इथे phloem आहे, इथे काय आहे xylem आहे, आणि याच्यामध्ये बघा 1 2 3 अशा सेम साईजच्या सेल दिसतात. याला काय म्हणतो आपण कॅम्बियम. So this is the meristematic tissue, and this tissue are responsible for the development purpose, growth purpose. So these are present in a dicot only because of the secondary growth is takes place. So we see here, it is a meristematic tissue present in between the xylem cells and phloem cells. So it is called as a open type of a vascular bundle. Why you call as open? Because of a presence of a cambium ring. It's also called as a fascicular cambium or vesicular cambium. Okay. The fascicular means what? In between in between two vascular bundles, there is a joining between joining of this bundle takes place by cambium. So that's called as a fascicular. And vascular cambium is vascular bundle मध्ये असेल तर त्याला vascular cambium. So is origin from the origin, this arranged or is originated from the procambium cells. Okay, आपण ज्यावेळेस आपण अपायकल मेरिस्टेम मध्ये बघितलेले the different types of a meristem here. So there is a procambium meristem cells are present, and these are the responsible for the development here. So these cells are made up of the parenchymatous cells. It is also having the thin wall cells having this dense cytoplasm and with having the prominent nucleus. After this secondary growth, dicots occur due to the cambium here. So due to the cambium, we call as the open vascular cambium here. So this is the structure of the vascular cambium here. Next here, that's the third, we call it the still region. So this is the still region. Okay. So we can observe the medullary ray. So in between the two vascular bundles, there is a part that is called as the medullary ray. So it is also called as a pith ray. This is occur between the vascular bundle composed of a parenchymatous cells are radial. If you see this one, some kinds of radial cells we can find out here. During this secondary growth, cells are medullary give rise a interfascicular cambium. या काय करतात? या दोन्ही व्हॅस्क्युलर बंडल्सला जॉईन करतात, म्हणून त्याला काय म्हणतो आपण इंटरफॅस्क्युलर कॅम्बियम. सो टू टाइपच्या कॅम्बियम, इंट्रा अँड इंटर. सो फंक्शन आहे रॅडियल ट्रान्सपोर्ट ऑफ वॉटर अँड प्रोव्हाइड द इंटरफॅस्क्युलर कॅम्बियम. इथं रॅडियल म्हणजे याला वॉटर सप्लाय करण्यासाठी ह्या मेडुलरी रेज काम करतात. So next here, fifth region, last one. So this is the center part here. So that center part of the stem is called as a pith here. It is also called as a medulla. This region here. So located towards the inner side of a vascular bundle. So this is the vascular bundle, and centered to the here there is a presence of this pith region. Extra central position of the stem. Mostly this pith region is made up of a parenchymatous tissue. So some kinds of hexagonal structure, loosely arranged, we can find out here. Sometimes thickening occur. जसं लिग्निफाईड होतं, सेकंडरी ग्रोथ वाढते, ज्या प्लांटची हाईट जास्त वाढते, तर हे लिग्निफाईड होत चालतात. ओके, त्यामुळे त्याची थिकनिंग ऑकर होते. द सेल्स ऑफ आउटर रिजन आर स्मॉलर व्हाईल इनर इज द लार्जर. तुम्ही इथे बघताय आउटर रिजनला म्हणजे अर्ली स्टेज आहे, आणि हा जे मिडल आहे लार्जर सेल्स आहे. So we can find out here. This is the pith parenchyma. So this is the center. So what is the function here? So that is called as the storage of a food material, or is some having some kinds of a pith cavity. Tell us about the pith cavity. So this is the books here. From this book we have taken this one material. त्याच्यामुळे काय problem? Hello? Any problem? So this is the
No,
dicot.
sir. No, sir.
Dicot structure. So hypodermis, the dicot stem is differentiated into following point. First is the epidermis, second is the cortex. Cortex are divided into again hypodermis, outer cortex, inner cortex, endodermis. After that, there is the presence of still region. This is the center region. This is composed of a pericycle, vascular bundle, medullary, and pith. So these are the internal organization.